You should get it as a handout, the 20 amino acids, okay? It'll help you. I'm not going to ask you to memorize them. The only one that's really, I would say, just super crucial to know is glycine, and I'll tell you why. The reason why I would say it's really important to know glycine is it's because it's the only one that has two identical groups. So instead of this group being what makes it distinct, being like a methyl group or a aromatic group or anything like that, it's another hydrogen. Okay, so this amino acid has two identical groups, which is the hydrogens. We also say that it is the only one that is achiral, which we didn't spend much time on earlier, so it's just kind of a weird thing that you might see in the book that it's going to say that it is achiral. So, a zwitterion. When we're talking about amino acids, it's common to talk about zwitterions, and this is something that is electrically neutral overall, but it contains a negatively and a positively charged group. So looking at glycine right here, we have the positively charged amino and the negatively charged carboxylic group, but overall the positive and the negative charge will cancel each other out, just like a proton and an electron canceled each other out, right, to give us a total of 0. Same thing here. So all amino acids, for the most part, are examples of, or can be examples of zwitterions. So the isoelectric point is the specific pH at which a given amino acid, so it could be different, but will exist in solution as a zwitterion. Okay? So the best way to understand this is to practice. So the first thing we're going to do is identify amino acids that contain the following. So we're going to go back to our table and we're going to look at that, and then we are going to practice the zwitterions, because I believe Sapling is going to have you practice on that. Okay, so this is going to be in class work. It's still September when I'm recording these. This is going to be in class work for November 18th. So A, we are looking for a hydroxyl group. If you remember back to our functional groups, these are OH groups. So looking at our table, I see none in the first group. Now looking at the second group, the one at the bottom, polar uncharged R groups, the bottom left, I see that serine has CH2OH in the blue box, SERINE. So I'm going to say serine. You could also say, if you look on the bottom right hand side, you could say tyrosine, which has the OH group, it's the middle one on the bottom right. So we could say serine or tyrosine. The second one says a branched hydrocarbon chain. So we're looking for something that is CH based, and then we're thinking branched, right? Not straight, but branched. So looking at this, we're looking for something that doesn't just go in a straight line. So I see in the upper left-hand corner we have valine and leucine. They are both branched hydrocarbons. And the last one says look for a protonated amine group. Okay, so that means since it's protonated, we are looking for an NH3 plus in the blue box. So because we're only focused on the side chains, which are the things that are in the blue box. So let's go back. And we're looking for a protonated amine group. So I don't see any in the upper left. I don't see any in the lower left. I don't see any in the bottom right or the group above that. But looking at the upper right-hand box, I see that lysine, arginine, and histidine all have a nitrogen that carries a +1 charge. So lysine, arginine, and histidine in the upper right-hand corner all have a nitrogen that carries a positive charge. They have other nitrogens that don't have a charge. Arginine, histidine, and lysine. Okay. So just kind of looking at the differences between the amino acids based on their side chains and those functional groups. So you have to think back to when we talked about organic and the different functional groups. All right, so this says, draw the structure of isoleucine and determine the charge on the molecule Na, and then it says acidic, neutral, and basic. So the first thing we have to do is draw isoleucine. So let's find isoleucine over here. So isoleucine is in the upper left-hand box. It has a branched hydrocarbon. So let's make sure we draw that correctly. Okay, so I'm going to draw Okay, so hopefully, looks like I got everything on there. So we have to draw it in an acidic solution, a neutral solution, and a basic solution. So we have to remember back to what it means to be an acidic solution. This is where we're going to have protons in excess, so everything that can take a proton will take it. So if we're looking at this particular structure right here, if we're in an acidic solution, this carboxylate group is going to want to take one of these protons, but there isn't a negative charge anywhere else on the structure to where we would need or could accept or donate a proton. Okay. Now, when we're talking about a neutral solution, probably should have done that one first, but this is where isoleucine is going to exist as a Zwitter ion. And you think back to what we said a Zwitter ion was. This is where both the negative and the positive charges will exist together, but then leave the molecule neutral overall. Okay, so this is the Zwitter ion. Now the last one is basic. So we have to think about when we did acid base, what we said happened with basic solutions. So in basic solutions, we are going to basically remove the H pluses, as many as we can. Okay, so if I draw this now, the nice thing is they are going to be somewhat related to each other. Okay, so if I start with the neutral form, so now I'm neutral, I've already taken away the proton from the COOH group, and that left me negative, so that happened at a neutral pH. So now if I'm going to draw the amino acid in the basic form, I'm not going to change that group right there because I've already removed the proton when I moved through the neutral pH. Instead, the only proton I could remove is the one that exists on the NH3+. Okay, so this particular proton is what gets removed. So minus that. When I go from neutral to acidic, I'm going to add a proton to that negative group. And it's like that for all of these amino acids. When you have an acidic solution, everything that has a negative group is going to accept a proton. And if you are in neutral, it's going to appear as it does on the table that I showed you in the book. And if once you read to the basic pH solution, then you're going to remove those protons from the carboxylic group or the amino group. Okay, Sapling's going to have you go through 2 of these. I suspect you're going to want to ask Dr. Morris or Dr. Spencer, whoever's there with you, to kind of go through those with you. And I think this is a great question to have them help you, because the more you see it, the more you start to see the pattern associated with this pH relationship and the structure of the amino acid. Now, you do not need to memorize the amino acids. Remember, the only one I'm asking you to know is glycine. Otherwise, I will give you the structure. Okay. So, one of the things that makes amino acids kind of unique is the fact that it's able to act as both an acid and a base. And we saw that up here. It's able to accept and donate protons, which is the definition of an acid and a base. So they have this ability because at a certain pH value, which is different for each amino acid, nearly all of the amino acids can exist as a Zwitter ion, which we've seen. If the acid is added to a solution containing the Zwitter ion, the carboxylate group captures the hydrogen and the amino acid becomes positively charged, which we've seen. If a base is added, the ion, there is the removal of the H plus ion from the amino group of the Zwitter ion, producing a negatively charged amino acid, which is what we've seen. Okay, so the most important thing here is to understand that the structure of the amino acid allows it to act as both an acid and a base. All right, so let's start building. Okay, the first thing we're going to build is what's called peptides. So peptides are going to be 2 or more amino acids joined together into chains. We join the amino acids together with what's called a peptide bond, so it's a lot like the glycosidic bond. And this is where you have an amide bond joining 2 amino acid units in a peptide or protein. A polypeptide is many, so a chain of about 50 or so amino acids. Okay, so we're going to start joining them together. So the peptide bond, if we think back to functional groups, okay, so that was one of our functional groups. So we have the amino, and then you have the carboxylic acid, and when they come together, they are going to form the amide bond, which is this group right here. Okay, and this would be amino acid number 2, and this is amino acid number 1, and that can just continue on and on and on and on. All right, so we said proteins are super important, and they are. We've already defined the fact that they're composed of high molar mass, mainly or nearly entirely composed of amino acid chains. There are 2 different groups of of proteins when it comes to structural classification, so what they look like. The first one is a fibrous protein. This is a protein that's elongated or fiber-like, and it's insoluble in water, so your hair, thank goodness, right? And then you have globular proteins, which is generally spherical in structure and soluble in water, so not your hair. There are different levels of protein structure. We're going to define them here, and then I'm going to show you some pictures from your textbook that are in color that will help you hopefully visualize this. So the different structures are primary, secondary, tertiary, and quaternary. Okay, they're also given the same terms like this. Okay, so that's primary, secondary, tertiary, quaternary. So the primary structure is the sequence of amino acids in the polypeptide chain or protein. So it's the sequence. The secondary is a fixed arrangement of the polypeptide backbone. The The tertiary starts to get three-dimensional, is the unique three-dimensional shape of a polypeptide chain as a whole. And the quaternary structure is the arrangement of multiple subunits in a protein. Okay, so let's take our definitions and look at them more specifically. So this is the primary structure of human insulin. You can see here it's just a chain of 21 amino acids, and then you have a chain here of 30 amino acids. So they're just follow the leader all the way around. Okay. So this is considered to be the primary structure, straight chain of amino acids. When we get to secondary structures, we have two different types. We have what's called the alpha helix. It's named such because it looks a lot like the DNA helix. And then we have what's called the beta pleated sheet. Okay, so we can see here now we have not just a straight line, but we have hydrogen bonding, which is allowing for additional stabilization in that secondary structure. And then in the beta pleated, you can see we have actually two primary structures right there. And then there's a third one, and then a fourth one. Okay, so it's kind of like Legos, right? This is if you have, you start adding the Legos together and make a bigger structure. So these are both secondary ones right here. Now when it comes to tertiary, we start looking at three-dimensional structures, and this is of insulin. So you can see it has that alpha helical structure right here, and while the broad arrows represent the beta pleated sheets. Okay, so these are the beta pleated sheets. Okay. And so this is the three-dimensional structure. So you can no longer even see the individual amino acids or even any of the fine details that we saw in the alpha and the beta structures. Okay. So there are four major what's called attractive interactions that determine the shape and stability of the tertiary structure of the proteins. So we're going to talk about these first before we talk about quaternary. So those four are ionic bonding, hydrogen bonding, disulfide linkage, and dispersion forces. Okay? So ionic bonding is bonding that results from the electrostatic attractions between positively and negatively charged groups. Okay? So it's an affinity. Hydrogen bonding, on the other hand, is when you have a highly electronegative oxygen or nitrogen that is in close proximity to a hydrogen that is coordinated to another oxygen or a nitrogen. Okay? So it's the interaction in the dotted line. Now the disulfide linkage is a covalent bond that forms by the oxidation and linkage of two sulfurs on two cysteine residues. So this is specific to cysteine, which is an amino acid. The last one is dispersion forces. This is a force caused by the instantaneous imbalance of electrons about a molecule. These tend to be our nonpolar groups. So like CH3 and CH3, they are not going to form the ionic bonding, they're not going to have hydrogen bonding, and they don't have sulfurs to do disulfide linkage. So their only interaction is dispersion forces. Okay, so let's see what that might look like. Okay, so we see here we have our interactions. Okay, so in this case, let me zoom in. Here I have a positively charged ammonia, or NH3, and an O minus. So this would be ionic. Here I have hydrogen bonding at B. At B is another hydrogen bonding. At D here I have dispersion forces, and the rings, it's also dispersion forces. I have ionic bonding over here at A. At C, this is my disulfide bridges. At B, it's hydrogen bonding, and then again at D, it's dispersion. Okay, so Sapling's going to ask you to practice this, and I also would like to practice what we would predict the two residues, what type of interaction they would have. Okay. So we're going to have to go back and forth. So we're going to compare valine and isoleucine. So this is in class. So valine and isoleucine. And we're only comparing the R groups. So valine, is, I'm going to zoom out now, is CH3 and CH3. And isoleucine is another R group. They're right next to each other, right in front of the screen. So the type of force that occurs between CH3 and CH3, we said, is dispersion. So asparagine and serine is our next one. Let's see if I can get this thing to... There we go. So we have asparagine and serine. They're in the bottom left-hand corner. I see a lot of hydrogens and oxygens and nitrogens. So anytime I see that combination, the first thing I'm going to think of in the blue box is hydrogen bonding. Because that's what the rule states, is that when I have OH and NH and hydrogens, it's going to be hydrogen bonding. The next one is glutamate and arginine. So looking at my box, in the upper right hand corner I have arginine. It has a positive charge on that nitrogen. The box right beneath it has glutamate, which has a negative charge on the carboxylate group right below it. So because I have a positive and a negative, I'm going to go ahead and say that this is going to be ionic bonding. Now the last one is tryptophan and proline. So let's go back and look. Tryptophan is in the bottom right hand corner. It's got the big bulky group. I can't remember the other one. And proline. And proline, so it's in the upper left hand corner. And you see in the blue box it's nothing but CH2, CH2, and CH2. So these are, again, another example of dispersion forces because it's carbon and hydrogens in that blue group, in the blue box. So the only example we didn't see of that tertiary stabilization is the disulfide bridging, but that's only between two cysteine residues. So the only time you would see it is if you had two cysteine residues. So Sapling is going to have questions on this, but having that amino acid table is going to be helpful because you'll be able to quickly reference what the amino acids are. All right, so the last structure is quaternary. This is an example of hemoglobin in its quaternary structure. Hemoglobin is the protein that transports oxygen throughout your body. The quaternary structure is very complex, and it's actually the only time that the protein is what we would consider to be active and functional. It is going to be in its quaternary state. Okay, anything other than that is considered to be denatured, and so it's not functional if it's in any other particular level. All right, so let's sit them side by side. We have amino acid residue, alpha helix, polypeptide chain, and then all the assembled subunits, and that's our primary, secondary, tertiary, and quaternary. Okay, again, this one being the functional active protein. Okay, so that is going to end the lecture today. We'll start off talking about denaturing of proteins in the next lecture, And then we'll spend some time talking about enzymes. So this is about 28 minutes in to the lecture, so it should leave you about 50 minutes to ask specific, or sorry, it should leave you 20 minutes to ask specific questions about the homework, if you have any. And then hopefully the majority of them are probably going to be about the amino acids and the Zwitter ions would be my guess. Okay? I hope that all is going well, and email me if you have any questions or concerns, anything I can help with, and I'm happy to try to help you, even if it's through email.